Our first speaker today is Dr. Jang Chao Zhao. He's a professor with the Animal Science Department in Fayetteville. His presentation is bovine respiratory, microbiome, and BRD.
Good afternoon, everyone. Thank you, Don, for this opportunity to share our research on bovine respiratory microbiome and BRD. BRD is one of the most problematic disease in the beef industry in the US. It is estimated that BRD can cause $1 billion of loss annually. So it's a big problem in the industry. Clinical signs of BRD include depression, nasal discharge, ocular discharge, coughing, gaunt appearance, fever, lung lesion, etc. And the diagnosis is mainly based on subjective observation. So therefore, limited sensitivity and specificity can cause unnecessary or delayed treatment of BRD. There are several factors that can affect the onset of BRD and the respiratory microbiome. This include the stress related to weaning, the stress related to the new environment and the new diet, host factors, and also commingling with other animals that contains the pathogens. So all these factors can affect the onset of BRD. Currently, the treatment of BRD mainly rely on different antibiotics, which can result in the generation and spread of antibiotic resistance. Although this disease has been in the field for several decades, the problem doesn't seem to be resolved. The morbidity and mortality rates have not decreased over the last 20 years. So it's still a big problem. I think there are two major reasons or two major problems for the slow progress in science or treatment of this disease. The first problem is that people mainly focus on four major bacterial pathogens, include Histophilus, Mycoplasma, Mehemia, Pasteurella. The thing is, these four opportunistic pathogens could also be isolated from both healthy and sick animals. So you can still see these pathogens or opportunistic pathogens in healthy animals. And little is known about the hundreds of other species. So there are a lot of bacterial species, not only these four pathogens. We don't know much about those other bacterial species in the respiratory system. That's one problem. The other problem is that nasopharynx is the major sampling site to study BRD. And we know this sample approach is kind of invasive. It requires a very long double-guided swab to collect the samples. And it is not practical or easy for a large epidemiological study. So this is another problem. So our objective or questions are to focus on the whole microbiome, including the temporal and spatial dynamics of the microbiome. That is all the bacterial species in the respiratory tract, not only the four bacterial pathogens. And also we are trying to answer if we can use a very easy and fast nasal swab approach to replace this invasive nasopharyngeal swab approach. So those are the two major objectives of this study. And this is the study design. We collected samples from both the upper and lower respiratory tract. The upper respiratory tract include this very easy nasal swab and also the traditional nasopharyngeal swab. Also, we included BAL from the lung. And we collect the samples from healthy animals. This figure shows each row represents one animal. And the top several calves never got BRD. And then we collect samples from these calves when they had BRD, and also collected a sample from a healthy control. This is kind of the study design and sample collection. We used this next generation sequencing approach, MiSeq, to study the whole microbiome. And we used bioinformatics and statistic ecological models to study the bone respiratory microbiome. And first, I'm going to show you the biogeography, that is the upper and lower respiratory tract microbiome. This figure shows the alpha diversity, which is panel A, and also beta diversity. So you can see the difference in their diversity and the relationship between the upper and lower respiratory microbiome. And panel C shows the top 15 bacterial species in the nasopharynx, in the lung. You can see those top 15 bacterial species are present in all the three niches, although the abundance are different. We were also able to identify the signatures for the three different niches. And also next, we used a technology we call random forest. It is one of the machine learning technology to predict or diagnose cows with BRD from the healthy ones. So I'm not going to the details of this technology, but the take-home message is that we can predict the onset of BRD using all the three different niches: nasal swab, nasopharyngeal swab, and BAL. And also we obtained the accuracy over 90%. You can see that from this area under the curve, all the three niches, we can predict the onset of BRD very accurately. And also we were able to identify several bacterial species that the differences are very consistent among the three niches. So that means we can predict the onset of BRD using nasal swab, which has at least as good a accuracy as nasopharyngeal or BAL. We also studied the changes of the respiratory microbiome before and during the onset of BRD, during the disease progression. And again, we were able to identify a set of bacteria that change in their abundance before and during the onset of BRD among the three niches very consistently. So that is another sign that we can use nasal swab to reflect or to replace nasopharyngeal. And lastly, we diagnosed the BRD using these samples collected from the upper and lower respiratory system. Again, we can predict we can diagnose the onset of BRD using all the three sampling approaches with a very high accuracy. Again, we also identified a number of bacterial species that change consistently among the three niches. So all this data shows that we can use nasal swab to replace the traditional invasive sampling approach of nasopharyngeal in regard of prediction, disease progression, and diagnosis of BRD. And also we were able to identify a panel of microbiome biomarkers for the prediction and the diagnosis of BRD from the NAIRS. And we have been able to apply for an international patent to protect this panel of biomarkers. And this study also allowed us to win a USDA grant to study the mechanisms, to study the interactions between the respiratory microbiome and the host. This study involves 3 research stations from the Arkansas, from UARK, including Basewell, Savoy, and Hope. Thanks Don and his group in Basewell, we have been able to finish the 2 animal trials, and now we are in the process to analyze the data. Finally, I want to thank my lab members. And also, this research has been mainly done by my postdoc, Dr. Jianmin Chai. And also, I want to thank my wonderful colleagues, Dr. Beth Kegley, Jeremy Powell, John Richardson, and Sarah Kapik. This is a list of funding agencies that have been supporting me during the last several years. And I also want to thank Don for his support of the matching grant and also his research group for several projects conducted in Basewell. And thank you all for your attention.